Assalamu alaikum. So this is the second lecture of the enzyme-based molecular techniques. In this lecture, we'll be talking about known as polymerase chain reaction. I know that many of you have been waiting for this lecture just to learn about what PCR is all about. And in fact, they're right. PCR has truly revolutionized the field of molecular biology and medicine as well. So what is polymerase chain reaction? So it's basically, from the name, it's a reaction, right? So it's a chemical reaction that is a chain reaction, meaning that متسلسل. So it goes one round and second round and third round and so on. And this reaction is catalyzed by the enzyme DNA polymerase. And the whole purpose of this reaction of PCR of this technique is to make lots of copies of a specific region of a DNA. So it's almost like cloning, except that it's much faster. It's very sensitive as well, and it has a lot of uses. Now, this technique was invented by a scientist known as Colin, and he became a millionaire with this technology, and he quit science actually. So, in order to catalyze this reaction, we need a number of components. We need the DNA template that we have to start with. We need a pair of primers, because a DNA polymerase cannot initiate DNA synthesis without primers. And these primers are DNA primers, by the way. Okay, so we don't need RNA primers like in DNA replication. So naturally in cells, DNA replication, DNA polymerase requires RNA primers, but in these reactions we can use DNA primers. Now we also need the enzyme itself, but this enzyme has to be heat stable enzyme, and we need its substrates, and they are the deoxyribonucleoside triphosphates. So we need all four of them. Note that what's missing is, if you compare this to the sequencing reaction, what's missing is the dideoxynucleotide. We don't need that. Okay, so the idea here is that each PCR reaction, the PCR reaction is made of cycles, so number of cycles, and the number of cycles is about 25 to 30 cycles. Each cycle is composed of 3 steps, and the steps are a denaturation step, okay, and this in this step we have separation of the 2 strands from each other, so we break up these strands, followed by another reaction, another step whereby the temperature goes down from 95 degrees all the way to 50 to 70 degrees. So it's variable. It's really a variable temperature depending on something that we'll talk about now. And then the temperature goes up to 72 degrees. This is the temperature, the optimal temperature of the DNA polymerase. So here we have separation of the 2 strands. Then we have the annealing temperature. This is where the primers bind to the DNA. And then we have the polymerization or the synthesis step at 72 degrees. Okay. Now, so what is that DNA polymerase, special DNA polymerase that we need? This DNA polymerase is known as Taq polymerase because it was isolated from a thermophilic bacteria known as Thermus aquaticus. So they took the T from Thermus and AQ from aquaticus. So from the name, it means that this bacteria lives in hot springs. Okay, so aqua in Latin means water, same thing in Spanish, and thermos from temperature. Now this enzyme is stable at 95 degrees. Of course, if we use a human enzyme, it wouldn't really survive. It would be denatured, it would become non-functional. Okay, but this enzyme can be stable, can stay stable at a temperature of 95 degrees amazingly, but its optimal the optimal temperature for the enzyme is 72 degrees. Okay, now, so these are the steps. First, we have the denaturation step, whereby the template is denatured. So the temperature goes up to 95 degrees. The two strands are separated from each other. Then the temperature goes down to about 50 to 70. Again, it's variable, so let's say 60 degrees, allowing the primers to bind to the DNA template. Now remember, I do not really have just one molecule of DNA. We could, by the way, okay, but we have many pieces of the DNA template. We also have a lot of primers. Okay, so some of these primers would anneal, that is hybridize or bind to regions of the DNA. These regions are flanking regions, meaning إنهم جناحين, meaning they are on the side. So they surround the region of DNA that I want to amplify. The temperature goes up to 72 degrees, allowing for the DNA polymerase to start DNA synthesis. Okay, so this again, that's the first cycle. The second cycle, we have denaturation again, annealing, primers bind to the DNA, and then we have DNA synthesis, and so on. So right at the third cycle, now we have the DNA size that we want, or the DNA fragment that we want. And then subsequently, we have further amplification of this region. And I'll show you right now of how it looks like. So we have about an average of 25 cycles when we do a PCR reaction. And this is sufficient to give us a large amount of DNA, okay, DNA product. So this is actually the instrument of how it looks like. This is a modern instrument. It's very efficient, really small. I do remember when I first did a PCR reaction when I was at your age, actually, in the US. The instrument was a bit larger, and the reaction or the whole cycle, PCR cycle, everything, the whole reaction took about 18 hours to finish. Nowadays, in like half an hour to an hour, we can really finish all 25 cycles. So it has become really good instrument. So after the DNA reaction is done, we take our sample and we run it on a gel. So we load the DNA in wells, just like seen in here. And after the DNA is run, we stain DNA with ethidium bromide or with whatever dye we use. And notice in these reactions, we have a number of samples in here. And notice that in each one of them, we only have one DNA band, okay? Meaning that we have only a single product. So DNA amplification is very specific. What does determine specificity of DNA reaction, of DNA amplification? The primers. The primers are the main determinants of where DNA is amplified exactly. Notice that we need both primers to surround the region of DNA that we want to amplify. Okay, so if you have only one of them that anneals or hybridizes to or binds to DNA, that's not enough. We need two of them to bind to amplify a specific region. Now, let's say it's very common that primers would go somewhere else and bind to or anneal to DNA. Okay, and we can prevent this from happening by choosing the right temperature, and that's why the temperature of the annealing step of any PCR cycle is variable. So it depends on the optimal point, the optimal temperature that specifies that or specifically allows primers to bind to DNA. Okay, so notice here, that at low temperature, for example, we have a number of products. If we increase the temperature slowly, eventually we would get this perfect temperature that allows primers to bind to DNA specifically, okay, allowing for a single product to be amplified. Now, how can we take advantage of that? Well, simply, why do primers go somewhere else on the DNA? It's because there is what we call homology. Homology means that there are similar sequences within the genome, and they are scattered. They are found in different places. Now, why do we have these homology or homologous sequences? We have them because sometimes we have gene families, like for example histones, the histone genes. We have histone 2A and 2B. The sequences, the DNA sequences of histones 2A and B are very similar to each other. They have high homology, as we say it, and that's why the primers for H2A may in fact bind or anneal to a region within the gene H2B, amplifying it as well. So what we can do is that we can really identify gene families by using primers at low temperature of the annealing step, allowing these primers to go somewhere else, amplifying it, and that helps us in the identification of genes within a family. So let's watch this animation video together. Okay, here we go.
A sample of chromosomal DNA, also called genomic DNA, can be used as the starting material for the polymerase chain reaction, or PCR. With PCR, an investigator can amplify a single copy of a DNA segment into billions of identical copies. The DNA segment of interest, or target DNA, is indicated in red. In addition to the target DNA, a PCR reaction contains several other ingredients. These include free nucleotides, DNA primers, and the enzyme Taq Polymerase. The primers are typically about 20 nucleotides long and are complementary in sequence to the ends of the target DNA. Taq polymerase is derived from hot springs bacteria and can tolerate the intense heat of a PCR reaction. A PCR reaction lasts several hours and typically consists of 20 to 35 repeating cycles. A cycle begins by heating the reaction mixture to 95 degrees Celsius. The heat denatures the DNA, breaking the hydrogen bonds that hold the strands together. After denaturing the DNA, the temperature is reduced to around 60 degrees so that the primers can form hydrogen bonds, or anneal, with their complementary sequences in the target DNA. Note that the primers and the target DNA follow base pairing rules. An adenine, A, pairs with a thymine, T, and a cytosine, C, pairs with a guanine, G. In the next phase, the temperature is raised to 72 degrees Celsius. Taq polymerase functions optimally at this temperature and begins polymerization, adding nucleotides to the 3' end of each primer attached to the DNA strand. After one complete cycle, there are 2 double-stranded copies of the target DNA. A sample of chromosomal DNA, also called genomic DNA, can be used as the starting material for the polymerase chain reaction, or PCR. With PCR, an investigator can amplify a single copy of a DNA segment into billions of identical copies. The DNA segment of interest, or target DNA, is indicated in red. In addition to the target DNA, a PCR reaction contains several other ingredients. These include free nucleotides, DNA primers, and the enzyme Taq Polymerase. The primers are typically about 20 nucleotides long and are complementary in sequence to the ends of the target DNA. Taq polymerase is derived from hot springs bacteria and can tolerate the intense heat of a PCR reaction. A PCR reaction lasts several hours and typically consists.
And this is it. Okay, so what do we use PCR for? Well, we use PCR to discover gene families, like I mentioned. We can use it for disease diagnosis. Okay, so for example, let's say that there is a deletion of a gene. We can try to amplify this gene in normal individual, and we would get a signal. Of course, we would get an amplified DNA product, or what we call an amplicon. Okay, so let's say that we want to amplify the same gene, but in a patient with this gene deleted, we would not be able to amplify this gene. Okay, or the amplicon would be much shorter than normal because part of it is deleted, and so on. So there are so many uses of PCR. We can use it as well for paternity and criminal cases as well. So instead of using RFLP, which takes a long time and it requires the use of radioactive probes, well, we can just do PCR to amplify certain regions in the DNA. These regions that could distinguish individuals from each other. Okay, so if you remember the genome lecture that you took previously, that there are individual differences in terms of number of repeats of VNTRs and STRs. Okay, we can also determine viral and bacterial load. What does that mean? Well, remember how I said that PCR is very specific? What does determine specificity? It's the primers. So the primers, we can design primers that are specific for viral DNA or bacterial DNA. Okay, so let's say that we suspect that someone is or has an infection, a bacterial infection. Okay, we want to confirm our diagnosis. So what do? We take a blood sample, we extract the DNA. Now, this DNA would be mainly human DNA, but we would have some bacterial DNA as well if this person is really infected with this bacteria. So what we do then is that using specific primers for the bacterial DNA, we amplify bacterial DNA. Okay? So we confirm the diagnosis that this person has a bacterial infection, and we know what the bacteria is, by the way, okay? Because the primers are specific for a certain type of bacteria or a certain strain. So what we mean by viral and bacterial load is we determine the quantity of bacteria that infect this individual, okay? And we can do this using quantitative PCR, okay? So we can use quantitative PCR. So how does that work? How does quantitative PCR work? Well, what we do is that one of the simplest methods is the use of Sybr green, okay? So what is Sybr? Sybr green is basically a small molecule that can bind to double-stranded DNA specifically. Okay, so it binds to double-stranded DNA. Once it binds to double-stranded DNA, it fluoresces. So if there is no binding between Sybr green and DNA, there is no fluorescence. But once it binds to DNA, it fluoresces, double-stranded DNA. Okay, so during PCR, what happens is that DNA is amplified, right? So if it in the denaturation stage, there is no fluorescence, primers bind, there is no fluorescence, and then we have DNA synthesis. Now, when DNA is amplified, what we have is that we have more Sybr green molecules bound to DNA, meaning that the amount of fluorescence that is produced is much higher. Okay, so with the next cycle, we would have, let's say, 4 DNA products. That means that we have more fluorescence that is emitted, produced from Sybr green, and so on. So with every cycle, we have more and more fluorescence that is detected. Okay, now the thing is, let's say that we take this sample right here. Okay, so this is what we see. At cycle numbers 2, 4, 6, 8, and 10, there's no signal detected. Why? Because, well, that's a limitation of the instrument itself. The amount of DNA is really very low for the instrument to detect the fluorescence that is emitted from Sybr green. Then at cycle 12, notice we have a signal. Now we have a signal because now the instrument can detect the signal that is emitted, produced from the DNA amplicon from Sybr green. Okay, so with every cycle, notice that the amount of signal, this is the amount of signal right here, so the amount of signal increases with every cycle until we get to a point where there is no more signal detected. It could be at cycles, notice at cycles 30 or 32. Why? Because the enzyme just got tired. Okay, maybe there is consumption of nucleotides and so on. Now, here in this chart right here, we have different samples. Okay, so we have this sample, the red sample is detected at cycle 12. Okay, but the green sample is detected at cycle 14. Now, why is that? It's because the green sample has a lower amount of initial DNA template. Notice here the purple sample. The purple sample is detected at cycle 18. Why? Again, because there is lower amount of the purple DNA to initially start the PCR reaction. Okay, now let's say that we have another sample right here that does not contain any, it does not produce any signal. It means that this sample is really negative. Okay, now why is it that the red DNA is detected first? Again, the initial concentration of DNA template is higher than all of the other ones. So let's say that we have in the red DNA, we have 1000 DNA templates to start the reaction with, versus the green sample that contains 100 DNA template molecules. So with the second cycle, the 10,000 become 20, the 1000 would become 2000, and the 100 would become 200. Okay, and let's say that the limit of detection is 10,000 copies. Okay, so now 10,000, I'm sorry, 1,000 becomes 2,000. 100 in the other reaction, the green reaction, becomes 200. 2,000 becomes 4,000 in the next cycle. Now the 200 becomes 400. Okay, now the 4,000 becomes 8,000, and the 400 becomes 800. Now, so far, nothing is detected because the limit of detection is 10,000 copies. Now, the next cycle, 8,000 becomes 16,000. Now we can detect DNA. We can detect a signal. Now, 800 becomes 1,600, 1,600. Still cannot be detected, and so on. So we detect the sample that contains higher amount of initial DNA versus a sample that contains lower amount of initial DNA template. So this is what we mean by a viral load. We can determine the quantity, the amount of viral particles or bacterial cells within infecting an individual, because we can really use sort of like a ruler, and we can have that, not an exact ruler. Okay, so we can really determine the amount of initial DNA template that we have in a sample. Okay. Now, eventually, by the way, the sample with the 100 molecules to start the PCR reaction with can eventually be detected later on in a later on cycles. So 1600 would become 3200, 3200 would become 6400, 6400 would become 12,800. Now it can be detected, okay, but later on in a cycle. So here, let's say we have 10 DNA molecules versus 100 DNA molecules to start the reaction with versus 1,000 versus 10,000 and so on. Okay, 100,000, 1 million DNA molecules. Of course, you can detect more DNA in here. Okay, or initially you can detect it earlier than this sample and that sample and that sample and so on. So this is what we mean by a viral load.